phospholipid. Okay, so phospholipid is grouped under lipids. Okay, so for the structure of phospholipid, as you can see from the diagram, it consists of a glycerol molecule. Okay, so a glycerol molecule, it has, sorry, okay, it has three carbons. Okay, so it has three carbons. So it has three carbons, okay, for the glycerol molecule. So two of the carbon is will be bonded to a fatty acid. Okay, so a fatty acid, one fatty acid molecule and another fatty acid molecule. So this fatty acid that is bonded to the glycerol will form the hydrophobic tail. Okay, so because why? Because as we have learned before, fatty acid mostly consists of hydrocarbons, carbons that are bonded to hydrogen atoms. Okay, so in this case here, so this type of fatty acid, it is saturated fatty acids as the chain is straight chains. Can so there's no presence of double bond. So when there's no presence of double bond, the molecule will be straight. But for the other fatty acid molecule, because it has double bond between the carbon, so it causes the formation of a kink. It causes the fatty acid molecule to be bent. Ini contoh je lah kan. So both can be saturated, both can be unsaturated, one can be saturated, one can be unsaturated, depends on the phospholipid molecule. Okay. And then okay, so the third carbon here, as you can see, it is bonded to a phosphate group. Okay, so as we can see from that diagram, the phosphate group, it has charge. It is negatively charged. So because it is negatively charged, it can attract water molecule. Okay, so it can attract water molecules. So because it can attract water molecule, so this will form the hydrophilic heads. Okay, the hydrophilic heads. And as you can see, there is another group which is the R. So if I'm not mistaken, you can refer, this is what we call it as the choline group. So in that choline group, it has a nitrogen atom that has positive charge. Okay, so because due to the presence of positive charge and also negative charge, this will form the hydrophilic heads. Okay, that has the affinity for water. Okay, affinity for water. This has this one, the hydrophobic tail does not have affinity towards water. Okay, and say this hydrophobic. Okay, so next, all right. So when phospholipids are added to water, so they're self-assembled into a bilayer. Okay, into a bilayer with the hydrophobic tails pointing towards the interior. Interior lah ni kan. So you have the phospholipid molecule and another phospholipid molecule between the two layers. So the hydrophobic tails will be interacting to one another through hydrophobic interactions. So this is hydrophobic interactions. So the hydrophobic tails will be interacting to one another, attracted to one another through hydrophobic interactions, and they are isolated from the surrounding aqueous solution. Okay, so if this is a cell, if this is a plasma membrane of a cell, okay, so these two regions can be, one can be the cytoplasmic side, the other one can be the extracellular fluid side, the outside of the cell. Okay, so both sides are aqueous, they have water. So the hydrophilic heads will be attracted to the water. Okay, okay. So the structure of phospholipid results in a bilayer arrangement found in cell membranes. Okay, cell membranes, and also other cells that are found forming the membranes of organelles, such as, for example, later in chapter 2 we are going to look at organelles, right? So there will be organelles that have membranes, such as the nucleus have membrane, the mitochondria also have membranes, the chloroplast also have membrane, as long as it is membranes. So the membranes are composed of phospholipid bilayer. Okay. So phospholipids are the major component of all cell membranes. So cell membranes, you can also term it as plasma membrane. Okay, cell membrane, plasma membrane is the same. Okay, membrane of the cell. Okay, so the next group is steroids. Okay, so steroids is also grouped under lipids. Okay, so steroids are lipid characterized by a carbon skeleton with 4 fused rings. So this is the main structure or skeleton of a steroid. It has 4 fused rings. Okay, 1, 2, 3, 4. Okay, 4 fused ring. So steroids are classified as lipid because they are insoluble in water. Why is it insoluble in water? Because as you can see, the structure, it has so much carbons and also hydrogens. They are hydrocarbons. Yeah, so because they are hydrocarbons, so therefore the molecule has less affinity. It is insoluble in water. Okay, it is hydrophobic molecule, right? So cholesterol. Okay, so cholesterol is an example of steroid. Okay, so cholesterol, as you will find later in chapter 3, it is also found embedded in the plasma membrane. It is also a component of plasma membrane in animal cell. Okay, cholesterol will not be found in plasma membrane of plant cell, but they are found in the plasma membrane of animal cell. Okay, so animal cell. So highlight this one. So tak ada cholesterol dalam plasma membrane plant of plant cell. Okay. So cholesterol belongs to the family of steroid because it shares similar chemical structure where it has 4 fused rings. Okay. So it is the precursor. What is precursor? The initial molecule to produce other steroids such as what do you call it, sex hormones, such as testosterone, estrogen, progesterone, cortisols, all the hormones that are made up of steroids. Initially, they are from cholesterol. Okay, so it is a precursor to vitamin D and for steroids hormones such as testosterone, progesterone, estrogen. These are sex hormone, and also cortisol. Cortisol is the hormone secreted by the adrenal gland. Okay, so if you don't know where is adrenal gland, you can search what is adrenal gland. So what is the function of cortisol? Cortisol is the hormone secreted by the adrenal gland. Okay. So estrogen, progesterone, testosterone are the hormones secreted by the gonads. Yeah, so search in your own time what is gonad. All right, gonads. Yeah, so gonads. Right. So these are the different types of steroids. Kan? So basically to produce all these steroid initially it is from cholesterol. Okay. So cholesterol, as you can see, they are embedded in the plasma membrane of animal cell only. Okay. So this is, as you can see, so this is the 2 layers of phospholipid molecule in a plasma membrane. So the way of how you are supposed to draw the presence of cholesterol is that it is located in between the phospholipid molecule. Okay, in between. Okay, as you can see, in between the phospholipid molecule. You cannot draw it somewhere in the middle like this. You cannot draw it protruding outside like that. Okay, so it is in between the phospholipid molecule, either on the top layer or on the bottom layer like that. Okay. So from the cholesterol molecule, it is the precursor to produce other steroids hormones. Kan? So we have cortisol here, we have the corticosterone, and then aldosterone, and then we have testosterone, estradiol, and progesterone. Okay, estradiol is estrogen, right? So these are the steroid hormones. So as you can see from the structures, the similarity is that all have 4 fused rings. Yeah, so they all have 4 fused rings, but the difference, as you can see, the difference between the steroids hormone is the yang paling obvious is the side chain here. Kan, the side chain is different. The side chain is different between one steroid to another steroid hormones. Okay. Same goes to the cholesterol. Asalnya macam ni kan, dia punya side chain dia. Alright. So between the different steroid hormones, they will have different structures, especially the one located here. Okay. Okay, so that's done with the phospholipid and also steroids. Do you have any question regarding on the on the two molecules, the phospholipid and also the steroids? Ada soalan? Faham?
Faham.
Faham, eh? Okay. So now we are going to look at the next subtopic, which is the nucleic acid, the last macromolecule. Okay, so the nucleic acids. So nucleic acid, basically they are polymers, which is, we call it as polynucleotides. Poly, banyak, kan, many nucleotides that are attached together. Kan? So polynucleotides. The monomer will be nucleotides. So when you attach one nucleotide to another nucleotide to another nucleotide, you form a polymer. We call it as polynucleotides. Okay, so you have to know the structure of a nucleotides. Okay, you have to know the structure of a nucleotides. So the structure of a nucleotide is like this. Okay, it is like this. So you have to know that it consists of a pentose sugar. Pentose sugar is a 5 carbon sugar. Again, a pentose sugar, 5 carbon sugar, and then the 5 carbon sugar is bonded to a phosphate group and a base. Okay, a nitrogenous base. Okay, a nitrogenous base. So that is the structure of a nucleotide. Nitrogenous base. Okay, so this nitrogenous base can be adenine, guanine, thymine, cytosine. Okay, so if this is the monomer for DNA, can. So if it's the monomer for RNA, another form of polynucleotide, so the bases will be either adenine, guanine, uracil, and cytosine. Okay, instead of having thymine, it has uracil. Okay, so that is the nitrogenous bases. And then for the sugar, there's two types of sugar. Either it is ribose or deoxyribose. So later we are going to look at what is the difference between ribose and also deoxyribose. All of you are required to know how to draw the structure of a nucleotides. Okay, so 5 carbon sugar bonded to a phosphate. The 5 carbon sugar also bonded to a base. Okay, so in this structure of a nucleotide, another thing that you have to take note is the number of the carbon. Okay, is the number of the carbon in the 5 carbon sugar. So again, you have to know how to label the carbons. Okay, so you look at this. So this is the pentose sugar. So how to number this carbon? So we start here. This is carbon number 1. Okay, carbon number 2, carbon number 3, carbon number 4, and up here is carbon number 5. Carbon number 5, it is bonded to the phosphate group. Okay, so carbon number 3 here, it has a hydroxy group. So this is really crucial for you to understand, because later we are going to look at how one nucleotide is attached to another nucleotides to form a polymer. Okay, so it says here the portion of a nucleotide without the phosphate group, we call it as the nucleoside. Okay, so nucleoside ini je lah. Dia ada base and also sugar. So that is nucleoside. So when added with phosphate, then only we call it as nucleotide. Okay, nucleoside, base and sugar. Adding the phosphate, then only we can call it as nucleotides. Macam tu. Betul lah. Okay, base and also sugar, right? Right. Okay, so now we are going to look at how one nucleotide is attached to another nucleotide and what is the name of the bond involved in attaching the two monomers together. Before, we have looked at, okay, in order for you to join one glucose to another glucose to form a maltose, for example, the bond between the glucose, we call it as glycosidic bonds. Kan? So, tapi kalau dalam amino acid, one amino acid to another amino acid is peptide bond. Okay, so now you are going to attach one nucleotides to another nucleotides, and the bond form, we call it as phosphodiester bond. Okay, one nucleotide attached to another nucleotide, the bond we call it as phosphodiester bond. Okay, so phosphodiester bond is a type of covalent bond. I've mentioned before in the carbohydrate parts, in the amino acids, in the protein parts, can when you attach two monomers together, it becomes a molecule. Okay, so the name of the bond, glycosidic bonds or peptide bonds, those are covalent bonds. Same goes to the phosphodiester bond. It is a type of covalent bond. So when it is a type of covalent bond, you cannot easily break the bond just applying heat. To break the bond, it has to undergo a series of reaction that involves maybe enzymes. Okay, the presence of enzymes, right? So okay. So a peptide, it is a type of covalent bond between adjacent nucleotides formed between the OH group on the 3' carbon of one nucleotide and the phosphate on the 5 prime carbon of the next nucleotide. Okay, for this one here, you have to look carefully. Okay, sekejap. Highlighter. Okay, so again, please, you have to know how to number the carbons of the sugar. Then only you can know how to number how the nucleotides are attached. Okay. Okay, kita guna biru. Alright. Okay, so this is the first nucleotides. Okay, the first nucleotides. Okay, the first nucleotides. So this is the second nucleotides. Okay, so first you have to know how to number the carbon. So this is carbon number 1, number 2, number 3, number 4, number 5. As you can see, carbon number 3, okay, of the earlier nucleotides or the previous nucleotides, okay, it has carbon number 3 as it has a hydroxyl group. Okay, it has a hydroxyl group. Kita dah belajar, in order for you to attach one monomer to another monomer, okay, the reaction involves is condensation reaction. Condensation reaction involves the removal of a water molecule. So where is that water molecule comes from? It comes from this reaction. So the 5 prime carbon where it has the hydroxy group will be removing its hydrogen atom. Okay, its hydrogen atom, and the next nucleotide that is going to be added, okay, at the phosphate group, okay, which is attached to carbon number 5, it has a, hydroxy group. Kan dia ada hydroxy group kat situ. So this hydroxy group will be removed. Kan? So remove hydrogen, remove hydroxyl, OH kat sini, OH. So it forms a water molecule. So once you have removed the hydrogen atom, the OH groups, then only you can attach carbon number 3 here to the phosphate. Okay, carbon number 3 to the phosphate. So you have here the bond will be formed between carbon number 3 of the previous nucleotide, okay, and the phosphate of the next nucleotide. Okay, so nanti kita akan tengok the polynucleotides, kan, the polynucleotide, they say lah, this is the polynucleotide, the one of the strands of DNA. Okay, so DNA is helical, kan, one of the strand. So the helical lah macam ni. So DNA has it consists of two polynucleotides that are coiled together, kan. So kalau if we were to separate the two strands apart, okay, so you get two DNA strands. You get two DNA. You get two polynucleotide strand. Okay. So this one here. So these two DNA strands, you have to know how to number the strand. The two DNA strands, it has directions. Okay, it has directions. So this one, at one end here it has 5. We call it as a 5 prime end. The other one is 3 prime end. Okay. So on the other end of the chain, it has a 3 prime end and the other one is 5 prime. Yeah, so maksudnya 5 ke 3, and also ini they will be complementary 3, 5. Okay, so the strands are, we call it as the DNA strand, we call it as anti-parallel. Okay, anti-parallel. Okay, so anti-parallel, what is anti-parallel? Okay, the top strand here, it runs from 5 to 3. The bottom strand will run from 5 to 3. Okay, so the two DNA strand, we call it as anti-parallel. Okay, anti-parallel. Why do we need to number the two strands? Why do we need to number the two strands? Okay, so for the polynucleotide, for example, awal start strand tu they start dengan 5 prime end. It starts with a nucleotide that has a 5 prime carbon bonded to its phosphate. Ini yang hujung, yang asal, yang awal. Okay, so along the strand, the last nucleotide of the strand, it has a 3 prime carbon. The last nucleotide. The first nucleotide, the 5 prime carbon will be bonded to its phosphate. It is free, doesn't bond to anything else. Okay, kat hujung lagi satu, dia ada nucleotides yang mana at the end it has a 3 prime carbon that it has a hydroxyl group. So by having a hydroxyl group at the 3' end, so what is the feature? At this end, you can add more nucleotides, more nucleotides, more nucleotides. Same goes to here. In order for you to add new nucleotides along the along the nucleotide chains, with the polynucleotide chains, you only can add the strand that has the label 3' end. Okay, 3' end. So I repeat, when you have a polynucleotide chain, okay, a polynucleotide chain, yang awal dia akan start dengan 5' carbon that has a 5' carbon that is bonded to its phosphate. Along the chain, along the polynucleotide, yang paling hujung, dia akan ada nucleotide that has a free 3' carbon with hydroxyl group. So by having this type of ends, that it has a 3' end, so it causes this last nucleotide of the chain to be able to accept another nucleotide, another nucleotide, and then another nucleotide, another nucleotide. You cannot add a new nucleotide at the 5' end of the chain. Tak boleh. Okay. Sebab apa? The 5' carbon is attached to its phosphate. It will not be involved in the condensation reaction. So the one that will involve in the condensation reaction is the last nucleotide of in that chain, because it has a hydroxy group, okay, that involved in the condensation reaction through the removal of this hydrogen to form a water, hydrogen, and also the OH of the phosphate. Okay. So because, as you can see, another thing that you have to take note here is that for a polynucleotides, for a polynucleotide, it has a backbone. Okay, it has a backbone. So what is a backbone? Repeating unit of phosphate sugar. Phosphate sugar. Phosphate sugar, phosphate sugar. We call it as the backbone. At the backbone of the polynucleotide, it has a phosphate that has negative charge. Okay, it has a phosphate group that has negative charge. So DNA, sifat DNA ni, it has, because it has negative charge, why does the phosphate has the negative charge? Originally, awak tengok asalnya dia dalam keadaan OH. So when the molecule or the nucleotide is dissolved in water, the phosphate will be ionized, the hydrogen atom will be released, and the O here, it becomes negatively charged. Okay, so that's why kalau dia punya macromolecule, kita namakan dia apa? DNA ataupun RNA, dia punya polymer dia. Deoxyribonucleic acid. Ribonucleic acid, due to the negative charge presence here. Why? Because it releases hydrogen ion into the solution, acidic condition outside. Okay, through the release of hydrogen ion. So that's why we call it as deoxyribonucleic acid. Ribonucleic acids. Okay, so DNA, dia punya property, the property of DNA or RNA, it is negatively charged. Okay, negatively charged. Okay. So it says here the chain of the nucleotides, the polynucleotides, has a direction. We call it as 5 to 3' orientation. Okay, 5 to 3' orientation, as I explained kat sini tadi. Okay, so the backbone is negatively charged due to a negative charge on each of the phosphates. Okay, so I emphasize again the bond between the nucleotides. Okay, the bond between the nucleotide, we call it as phosphodiester bond. Right, so the bond will be formed between carbon number 3 of the previous nucleotide in the chain and the phosphate group attached to carbon number 5 of the nucleotide that you are going to be adding to the chain. Okay. Okay, so okay, this one here, if you have this slide, okay, please remove this slide. Remove this slide. Supposedly, the labeling of this phosphodiester bond is incorrect. Okay, incorrect. Supposedly the phosphodiester bond, the way of how you label the bond, it is between carbon number 3 and also just before carbon number 5. Like that. Okay. Kalau you all extend sampai ke sini, dia jadi salah. It must be before carbon number 3, okay, including the phosphate, and before carbon number 5. Alright. So the nota yang sepatutnya saya dah update, dah upload dalam UFuture. Okay. So sepatutnya kalau you all dah download nota yang recent, saya dah re-upload. Saya dah tukar slide dia. Okay, it's supposed to be like this. Okay, like that. Okay, so this is one nucleotide. Sorry. Okay, so this is one nucleotide. Okay, so this is another nucleotide attached to the chain. Okay, so the bond for the phosphodiester bond, it is between carbon number 3 of the previous nucleotide, the phosphate, and before carbon number 5 of the next nucleotide. Okay, so again, I'd like to emphasize the directionality of the polynucleotide. You have to know the direction. Why is it 5 to 3? And the direction is supposed to be any DNA or RNA molecule. The direction is in the direction of a polynucleotides. Okay, it must start with a 5 prime and ends with a 3 prime end. Okay, start this sebab apa? Carbon number 1, number 2, number 3, number 4, number 5 is attached to its own phosphate. Yang awal. Okay, dia start dengan carbon that is labeled as 5 prime. Okay, and then the next nucleotide, this is carbon number 1, number 2, number 3, number 4, number 5. Number 3 here, it has it is attached to hydroxy group. Okay, because it is attached to the hydroxy group, it involves in the reaction of condensation. Okay, condensation that removes a water molecule later, kan, right? So hydrogen dia akan ambil, keluarkan OH daripada phosphate will be removed also to form a water molecule. Okay, so this is the backbone, repeating unit of phosphate sugar, kan. So the backbone, at the phosphate it has negative charge, so making the DNA molecule or RNA molecule negatively charged. Kan, negatively charged sebab apa? Yang dekat phosphate tadi ada O, yang asal dia adalah H. Asal ada O, asal dia adalah H. So when it is ionized, the hydrogen will be removed and the oxygen will become negatively charged. Okay. Yang ini apa dia? This one is base. Kan? So base, if it's a DNA, it can be adenine, it can be thymine. Okay. So if it's RNA, instead of thymine, it will be uracil. Okay. Okay, so next is the nitrogenous bases. So two families of nitrogenous bases. We have here the pyrimidines. Okay, the pyrimidines. So pyrimidine consists of cytosine, thymine, and also uracil. Okay, so this is the structure of cytosine, thymine, and also uracil. Thymine are only found in DNA. Uracil are only found in RNA. Cytosine found in both DNA and also RNA. So as you can see from the structure, pyrimidine only consists of one ring, okay, 6-membered rings. As for the purines, purine, it has adenine and guanine. It is a 6-membered ring, 1, 2, 3, 4, 5, 6, fused with a 5-membered ring. So purines, it consists of adenine and guanine. Okay, adenine and also guanine. Okay, so here we have nucleic acids. Okay, nucleic acids. There are two types of nucleic acids. We have DNA, deoxyribonucleic acid, ribonucleic acid. Okay, acid. And because DNA RNA is negatively charged, okay, and when it is in aqueous solution, the O will be ionized, okay, releasing hydrogen ion into the solution, into the surrounding aqueous solution. Okay, so deoxyribonucleic acid, deoxyribonucleic acid, because the sugar of DNA nucleotides, it consists of deoxyribose. The sugar is here, deoxyribose sugar in DNA. Kalau RNA, the 5 carbon sugar is ribose. What is the difference between deoxy and deoxyribose and ribose? You look at carbon number 2. Carbon number 2. Carbon number 2 of ribose, it has hydroxyl group. For DNA, at carbon number 2, it only has hydrogen atom. Okay. Deoxyribonucleic acid, deoxy, removes the oxygen, left with only hydrogen. Okay, deoxyribonucleic acid. Okay. Okay, so this is for the sugar in DNA and also RNA. Kita pause sekejap ya kelas. Okay, so what have I drawn here is two nucleotides. Okay, so the two nucleotides will be attached to one another, okay, through the process of condensation. So when it involves condensation, so it means that a water molecule has to be removed. Can so again, this I draw balik semula so that you all faham. So hydrogen removed, OH in the phosphate will be removed. Can OH removed. So this becomes a water molecule. Okay. So therefore, the bond forms will be between carbon number three, and also phosphate of the next nucleotide. So here, okay, we just erase out this, and then we just cantumkan je lah. Kan? Sempurnakan dia. So the bond between carbon number 3 phosphate that is attached to carbon number 5, we call it as the phosphodiester bond. So labeling dia mesti kat sini saja, antara sini sampai sini sahaja. That is phosphodiester bond. If you were to like terlajak sikit kan, contohnya macam ambil lajak sikit, lebih pada carbon number 3 and then lebih pada carbon number 5 here, so your answer would be wrong. Let's say if the question asks you to label the phosphodiester bond, so you have to label within the range carbon number 3 and before carbon number 5. That is phosphodiester bond. Okay, so this is how you attach the two nucleotide in a polynucleotide chain, either for DNA or RNA. Okay, so this is for one strand, right? So and then if I were like, for example, to add the next nucleotide, so saya draw dia warna hijau, let's say for example. So this is the next nucleotide. It has a base, sorry, a sugar, kan, a sugar. And then it said the next base is uh, thymine. Okay, thymine. So carbon number 5 bonded to its phosphate. Okay, bonded to its phosphate. So you just remove the OH. And you just attach the two nucleotides together. Yeah, so between carbon number 5, sorry, carbon number 3 and carbon number 5, kan? Okay, between carbon number 3 and also carbon number 5. Sekejap dia lagging sikit. Okay, so this is for one strand. So as you can see for this strand here, so it starts with a 5 prime and ends with a 3 prime. 3 prime, it has a hydroxyl group. Okay, a hydroxyl group. This is for one strand. So if this is a DNA molecule, it has to have a complementary strand. Okay, a complementary strand. So the complementary strand will run in the opposite direction, anti-parallel. So let's say for example, okay, anti-parallel. So ini dia macam mencabar sikit untuk draw. Okay, so let's say adenine, it has to pair with thymine. Adenine, thymine, okay. Pairing between the bases is adenine with thymine, guanine with cytosine. Okay, so kat sini because this is adenine, the base of the complementary DNA strand, it has to be a thymine. So thymine saya draw tadi warna hijau kan. So thymine lah kat sini, thymine. Okay, so this one, because the strand is anti-parallel, as you can see, so this one is, you have to draw it like terbalik. The sugar is facing this way. Macam tu. Okay, macam tu. So this is carbon number 1, number 2, number 2, number 3. This is carbon number 3 yang mana dia ada OH. Okay, so carbon number 3, it has the OH. This is carbon number 3. Number 4, yang number 5 ni, it is bonded to its phosphate group. Okay, so between adenine and thymine, between the two DNA strands, the two DNA strands are attached through the formation of hydrogen bonds. Okay, so hydrogen bonds between adenine and thymine, there will be two hydrogen bonds. Yeah, two hydrogen bonds. Okay, two hydrogen bonds. Hydrogen bond you have to draw using dotted line now. Okay, dotted line. Okay, so two hydrogen bond. Adenine and thymine, two hydrogen bonds. Okay, so and then okay, so the next strand is the nucleotide guanine pair with cytosine. So cytosine maybe saya buat warna purple. Cytosine, the base, okay, and then base is bonded to the 5 carbon sugar. Yeah, so draw dia terbalik. Carbon number 4, number 5, and is phosphate. Okay, so carbon number 1, number 2, number 3, number 4, number 5. 5 with its phosphate. So kat sini ikatan dia adalah antara carbon number 3 and phosphate of carbon number 5 of the previous nucleotide. Macam itulah, okay. So yang ni, this one thymine pair with adenine. So adenine saya draw dia warna merah. Base dia kan, the nucleotide structure bonded to a 5 carbon sugar, ada oksigen kat sini. and then the phosphate. Okay, so adenine, so this is carbon number 1, number 2, number 3, number 4, number 5. Okay, so the phosphoester bond is between phosphate and carbon number 3. Okay, so between guanine and cytosine, the attachment or the attraction between the two bases is through the formation of 3 hydrogen bonds. Satu, dua, tiga. Ada 3 hydrogen bonds between guanine and also cytosine. Okay, thymine, adenine, again 2 hydrogen bonds. So that's why DNA, the direction of the strand is 5, 5 to 3. The direction is 5 to 3. For you to add the next nucleotide, you only can add to the nucleotide that has exposed carbon number 3. For this one here, the strand runs in the opposite direction, anti-parallel. 5 ke 3. From 5 prime to 3 prime direction. So 5 to 3 prime direction. For you to add the next nucleotide, you add here at the 3 prime end. Okay. So that's why DNA dia punya orientation 5 to 3, and then the other one 5 to 3, anti-parallel. Okay, anti-parallel. So I hope that you know how to draw each individual nucleotide so that you know how to attach the nucleotides together. Okay. So next one. Sikit lagi, kita habiskan sikit je lagi. You all tak ada kelas kan? I want to finish off this chapter so that on Friday we can proceed with the next chapter. Okay. So this one here, there's 2 types of nucleic acid, the DNA and also the RNA. DNA provides the direction for its own replication. Genes in DNA directs the synthesis of mRNA and through mRNA control protein synthesis. Okay, our cell, inside our cell there will be nucleus. Inside the nucleus there will be the DNA. So inside the DNA there will be genetic codes. We call it as gene. So each individual gene will code for the synthesis of protein, kan? Untuk nak menghasilkan protein mesti berdasarkan kod yang ada pada DNA. Tapi DNA tu, the DNA molecule is really large molecule. So how to get a small portion of that information from DNA in order for you just to use that region to synthesize the protein from that gene encoded. Okay, so from the gene in the DNA, you are going to synthesize a smaller molecule which is the RNA. Kan? So RNA copies the information from DNA into a smaller molecule. So RNA will be single stranded because the RNA is copying the information from DNA. We call it as messenger RNA. Okay, messenger RNA, mRNA. So this mRNA will go to the cytoplasm to synthesize protein. So this protein can be enzymes, it can be hormones, or it can be any other proteins that the cell requires. Okay. So DNA. Okay, DNA structure. Maybe saya dah explain. Okay, so DNA structure, the two polynucleotide strand will be coiled. Okay, so the two DNA strand will be coiled together to form a double helix. Okay, a double helix. Okay, double helix. So double helix, the two backbones of DNA, double helix run in the opposite direction, 5 to 3, and then 5 to 3, opposite direction. Okay, so we call it as anti-parallel. The two strands of DNA is anti-parallel to one another. As you can see, the two strands are held by hydrogen bonds. Okay, so adenine with thymine, guanine with cytosine. Okay, so and then this explains on the complementary pairing. We call it as complementary base pairing. So if one strand it has adenine, the other strand will be thymine. If one strand has guanine, the other strand will have cytosine. The bond between the bases, adenine and thymine will have 2 hydrogen bonds. Guanine and cytosine will have 3 hydrogen bonds. This one you have to know. Okay, you have to know. You have to memorize. Okay. So nampak? Thymine and adenine 2 hydrogen bonds. Okay, guanine and cytosine 3 hydrogen bonds. Pernah ditanya untuk you all label kan the formation of hydrogen bonds between the two bases in a DNA strands. Okay. So this is the structure of DNA molecule. DNA molecule is double helix, can coiled together. As for RNA, RNA we have three types. Yang you all belajar, which is mRNA, and then we have tRNA, and then we have rRNA. Okay, rRNA. mRNA is messenger RNA. It copies information from the large DNA molecule. It's just one a portion of that gene. Kan? So mRNA copies information from a gene, and that mRNA will go to the cytoplasm to synthesize protein. In order to synthesize protein, the synthesis of protein will occur in the ribosome. Okay, ribosome is organelle in the cytoplasm. Awak dah belajar ribosome adalah organelle yang terlibat dalam protein synthesis. Okay. So in order to form that ribosome, the ribosome is composed of protein and also RNA, ribosomal RNA. So rRNA is ribosomal RNA, the component within the ribosome. Okay, and then we have tRNA, transfer RNA. What is transfer RNA? The function of transfer RNA is that it brings specific amino acid according to the code on the mRNA. Macam itulah. And so we have three types of RNA. Okay, so RNA basically it is single strand. Okay, single strand. But as you can see for this tRNA, boleh cakap madam, tapi dia macam ada double strand je. Dia sebenarnya satu strand that is folded into a shape, we call it as clover leaf shape. Dia fold akan ada base pairing lah. Yeah, so it is actually a single strand that is folded into specific shape to form the tRNA. Okay, so in this table here, it shows you the differences between RNA and also DNA. Okay, so as you can see from this table, we know that DNA is double-stranded. Okay, double-stranded, which is the two strands runs anti-parallel to one another, and the molecule is helical. Okay, coiled. And as for the RNA, it consists only single strands, much shorter because the function is just to copy the information from the large molecule of DNA. And then the pentose sugar for RNA is ribose. And then for the DNA, the pentose sugar is deoxyribose. Okay. And then for the RNA, the bases again consists of adenine, guanine, cytosine, and uracil. Okay. For the DNA, the bases consists of adenine, guanine, thymine, and cytosine. Okay, thymine. And then for the DNA, they are found in the nucleus, and also there are also DNA in the mitochondria and also chloroplast. Small amount of DNA in the mitochondria and chloroplast. And then for the RNA is produced in the nucleus, but the function of RNA, it functions in the cytoplasm. Okay, it functions in the cytoplasm. So therefore, RNA can be found both in the nucleus and also cytoplasm. Right? And then for the DNA, RNA, so there's only one type of DNA, the DNA that is located or stored in your nucleus. But for the RNA, there's three types: messenger RNA, transfer RNA, and also ribosomal RNA. For DNA, the molecule is much more larger because it contains all your genetic information. And as for the RNA, the molecule is much more smaller as they are required to synthesize protein. Yeah, they are required to synthesize protein. They only copies a small amount of information from the DNA. And then what else? DNA nucleotide sequence may encode gene. So each gene in DNA is a unit of genetic information. Each genes in the DNA can code for the synthesis of certain protein. For example, here let's say we have a large molecule of DNA. Okay, so large molecule of DNA, so this large molecule of DNA, it can be divided into several regions. So each region, we call it as gene. Okay, so each of these gene will code for the synthesis of specific protein. So let's say for example, this is gene 1 to it code for the synthesis of enzyme 1, for example. Okay, and then the third gene here, it is to synthesize enzyme 3, for example. Okay, so those are the information that is stored in your DNA sebagai storage saja, tapi information tu perlu di copy in smaller form, which is the RNA, so that the smaller form of information will be carried into the cytoplasm to synthesize the actual protein. Okay, So does not act as a gene important in the production of protein, right? So that's all for chapter 3. So we have covered all the macromolecules. We have covered from the beginning, which is the different types of chemical bonds, the functional groups, the on the water parts, okay, and on all the different macromolecules. So I want you to go through your notes, okay, go through each parts of your notes and do a mind map on each of those that component we have looked at for this chapter, kan. So if you uh, do a mind map, so it is much more clearer for you to understand on this topic. Sebab topik ni sangat besar, dia banyak benda yang you all kena fahamkan. So do a mind map, extract out all the information that are important in order for to help you to understand better for this chapter. Okay. So, do you have any question regarding what we have looked at for this lecture today? Saya tahu kita punya kelas ni agak petang, okay? Dan saya tahu bila kita dah masuk kepada nucleic acids, nak gabung-gabungkan nukleotide dengan 5, 3 bagai semua tu, it is hard for you to digest. Okay, so if you find today's lecture is a bit hard for you to digest, that's why kita ada SLT tu. Kan, saya cakap hari tu 1 jam lecture bersamaan dengan 2 jam your own revision. So please find time to look back on what we have learned today to enhance your understanding on today's lecture. Especially yang part label 5-3 tu tadi dengan formation of phosphorester bond tadi. Okay, so kalau tak ada apa-apa soalan, I guess we can end our session class. Okay, so thank you Ajab. Saya akan upload. Kalau saya lambat upload, you just boleh godek dalam tu daripada lecture sem-sem lepas pun ada juga. Okay, yang last week je, saya tak sempat nak upload lagi. Okay kelas? Tapi saya akan upload. I will upload the lecture. Okay, so itu je lah. Okay, class.
Thank you, madam.
Right.
Thank you, madam. Thank you, madam.
Okay.
Thank you, madam.
Alright.